Welcome to our podcast on the muscular system. This is probably going to be one of our shortest units and shortest podcasts of the semester, but that doesn't mean it's any less significant. We talked about the skeletal system in the last podcast, and without the muscles, really our skeletal system would be quite useless. So as we begin looking at the precursors of muscle cells, we're going to start again in our periaxial mesoderm, in our somites. We saw that we divided our somite into the sclerotome, which we have already mentioned from the previous unit, forms the vertebrae and the ribs, and the overlying dermomyotome. Now, the dermomyotome we're going to divide into separate divisions. The one closest to the skin is going to be the dermatome, and this is going to form the connective tissue elements of the skin and the dermis. But then the more medial between the dermatome and the sclerotome, this is the myotome, and this is where we form our muscle precursor cells. Now, we're going to spend some time talking about this process of myogenesis, just like we talked about osteogenesis and chondrogenesis. But in this case, these mesodermal cells and these pluripotential mesenchymal cells, we're going to induce them down a pathway of muscle formation to become myoblasts. Now, this is not like chondroblasts, chondrocytes that are going to be myocytes and osteocytes. What we're going to do with our myocytes, particularly with our skeletal muscle, and that's what we're going to predominantly be talking about, these myoblasts are going to be induced by a huge number of regulatory factors, and we are actually going to cover some slides that are in the most brand new textbook, not in yours, and we'll get to those in a bit. But what we're going to see is that all of these factors and inductive forces are going to impact upon this transcription factor, and this is called MyoD. This is going to regulate the transcription of muscle-specific genes, and it's one of many factors that are called myogenic regulatory factors, or MRFs. Now, as we impact our mesenchymal cells and head them down the specification pathway for myoblast, we're going to see that our myoblasts will eventually fuse into elongated columns that we refer to as myotubes. Now, we know in adult skeletal muscle, we have multinucleated, elongated myofibers or muscle cells, and this is where we get that multinucleation. As many myoblasts that are singly nucleated fuse together, that gives us our myotubes. Now, as we continue to be under the influence of our regulatory factors and we begin to express muscle-specific genes, certainly we know those are actin and myosin, or as we say in physiology, thick myosin and thin actin. Those are the myofilaments, and once we put all those into the contractile elements, form the sarcomeres, now we have a mature muscle cell, which we call a muscle fiber. So just putting this in a small outline, we can see our mesodermal progenitor cells, our mesenchymal cells, that will go through the process of determination into our myoblast. And here we can see one of those MRFs, the MyoD gene. We have another myogenic regulatory factor called MIF5. These are some of the earliest to ever be identified. Once we begin to express these MRFs, that's going to lead to differentiation. And once we begin to form our myotubes, then we see other regulatory factors turned on, such as myogenin. And then we undergo the maturation process, and we have our muscle-specific genes, again, such as actin and myosin, troponin, tropomyosin, dystrophin, all of those elements that we studied in the microscopic anatomy of muscle cells. But I want to look at some of this newer information as we begin to put this pattern together. We're going to go back and look at the influence of surrounding tissues, inductive influences on the myotome itself and really on the somite. In this newest illustration that we have from our textbook, we're going to see that there are both positive and negative influencing forces on this tissue that's going to give rise to our muscle precursors. We can see we have the neural tube, we have the notochord, and then we have our lateral plate mesoderm that's here as LPM, and here we have our somite, sclerotome, and the dermomyotome. This is the overlying ectoderm that they have shown in E. And from all of the literature that's been accumulating, putting this together in this picture, we see that the dorsal part of the neural tube, as well as the overlying ectoderm. They've painted these orange because these are going to have positive influences. And these are going to express, again, that Wnt family that we talked about back in our skeletal chapter, and bone morphogenetic protein. Both of these are going to be positive inducers of that myogenic pathway and of developing the myotome from our somite. We're going to see that the notochord and even this ventral part of the neural tube that we've shown in green, they are also going to have a positive influence. They are going to be secreting sonic hedgehog. Remember, we saw Indian hedgehog before. Well, here's sonic hedgehog again, and it's going to have a positive influence on the sclerotome. Now, this is going to help to distinguish our myotome from the sclerotome. However, if we look more closely at our lateral plate mesoderm, we're going to see that this actually has an inhibitory effect. So it's a negative inducer. It's going to keep the sclerotome tissue and the dermal myotome tissue intact until you reach a threshold level of the influences of Wnt and BMP to keep these cells from developing and migrating too rapidly. And these influences are going to be BMP4 again, which almost doesn't make sense. But if we're having too high a level of BMP4, that might be a negative influence versus lower levels, more positive. But then we also have the influence of fibroblast growth factor 5. These are going to be responding to different parts of our dermal myotome. And we can see that we have differential expression of these two BMP4 binding proteins, noggin and follistatin. It's possible BMP4, when it's expressed in the presence of noggin and follistatin, will have a positive influence, whereas BMP4 without those binding proteins on this side of the dermal myotome will have more of an inhibitory influence. But that's sort of the state of the art that we know now as far as the inductive forces that we see here. Wnt's, BMP, sonic hedgehog, and fibroblast growth factor playing a role on this somatic tissue. Now, once we form these, and especially with our sonic hedgehogs and Wnt's and the BMPs, we're going to basically begin to express MyoD, and we already saw that was a myogenic regulatory factor. MIF5 is going to be another one of those myogenic regulatory factors. And this new one, PAX3, again, different portions of the dermal myotome. And once we do this, we are going to begin to get migrating muscle precursors, migrating down into this area where we're going to start to form the musculature of our limb bud. And we'll talk more about limb bud formation in the last chapter we're going to cover in chapter 16. But we can continue to see the expression of PAX3. Another to mention here, and this is brand new, in c-Met. Apparently this transcription factor is going to be critical to change these muscle precursors from more stationary cells into migrating cells and allow them to migrate into the limb bud. We have a lot more genes that are being expressed that have been identified. We don't have roles for all of these yet. Right now we're just correlating the expression of certain genes, their locations in the embryo, and some of them, like c-Met, we're beginning to understand the roles of these as it relates to migration, differentiation, and development as a whole. Now, back to our embryo as a whole. When we look at our myotomes, remember, they're going to be part of these paired segments of mesoderm, the paraxial mesoderm called somites, and we see that arrangement. Well, as these myotomes begin to grow, we're going to be able to essentially break them down into the darker colors, which will give rise to our dorsal musculature, and the lighter colors, which is our ventral musculature. And if you think back to comparative anatomy with the lamprey, eel, and the shark, these are our epaxial muscles. These are our hypaxial muscles. Now, we don't have a lateral line like the shark to really help distinguish that. But we'll see in the next slide an image where we clearly see the delineation between our dorsal muscles and our ventral muscles. Now, our epaxial muscles, here's for instance of our epaxial muscles, the extensor muscles of our neck, muscles of our vertebral column, and we can see those again in that darker color. And our hypaxial muscles, these are going to be the ventral flexor muscles of our ventral vertebral column. Here in our cross section, we can pretty clearly see some of these short muscles in the back. These are going to be our epaxial muscles, epiaxis, if you will, above the axis, or dorsal. And here we have our hypaxial muscles, our lateral wall muscles, rectus abdominis. These are going to be the hypaxial muscles. We can see again some of these here, external obliques, rectus abdominis versus the smaller muscles associated with our vertebral column. Now, one thing to point out, I'm not sure if you were aware of this in the previous slide. You may want to go back and look at that again. But there are 3 preotic myotomes. And these preotic myotomes are going to give rise to the muscles of the eye, these extrinsic muscles that are going to help to move the eye in very fine detail and direction to help us really be able to focus our attention for our vision on very, very specific areas. These are some of the exceptions to just the regular myotome throughout the vertebral column. These are the most cranial, if you will, and they're going to serve the eye muscles. Now that was all about skeletal muscle. We have 2 other types of muscles, smooth and cardiac muscle, and both of these are going to be derived primarily from splanchnic mesoderm. Now for our cardiac muscle in our chapter 13, we already went into all the different tissues that are used to derive the heart, so we're not going to expand on that here. We know we have splanchnic mesoderm. We know we have neural crest cells. So just to distinguish what we're doing in smooth and cardiac muscle from skeletal, skeletal is going to be somatic from the somites. Smooth is going to be splanchnic and cardiac. We refer to chapter 13 for those different components that are contributing to our heart. Now, really, that's a big global overview of skeletal development. We'll put a little bit more of it together as we develop our limb in the next chapter. But we can, in fact, have some anomalies present with the muscles. It's not uncommon to have the absence of one or more skeletal muscles. And in this case, we have a syndrome. It's called Poland syndrome. And this is going to be the absence of the sternal portion of the pectoralis major. Now, again, we have correlations with things. I don't know that we really know the cause and effect of many of these things. But with Poland syndrome, not only do you have the absence of that portion of the pec muscle, but you also have syndactyly. And syndactyly is going to be fusion of your digits, your fingers. So you're not going to necessarily see very distinct digits on your hand. We had apoptosis did not occur appropriately, and so we didn't separate the digits. Some other anomalies, and these are progressively more and more severe. Arthrogryposis multiplex congenita. This is a very extreme problem, widespread failure of normal muscle development. And this is really going to lead to the inability to move. And oftentimes when you see infants born with this, they are very contorted, their bodies are misshapen, and really quality of life here is 0 to none. This is an extremely debilitating problem. Another severe problem, we talked about congenital diaphragmatic hernias some time ago. Well, this is congenital absence of the diaphragm. And as you can imagine, we are going to have pulmonary hypoplasia because now our abdominal contents are completely pushed up into our thoracic cavity. Very insufficient room for the lungs to develop, and this is going to cause extreme problems, especially with breathing. Now, the last area to really cover is something that we've probably most heard of and most familiar with. This is congenital muscular dystrophy. The onset of this disease certainly is at birth, it being congenital, and we see the generalized muscle weakness, joint deformities, and the image that you see here is what we typically see on Labor Day, one of Jerry's kids with the telethon. And so this degree progresses very slowly. There are several different forms of this. And the Fukuyama form of this congenital muscular dystrophy is much more severe, and often it will include some mental deficiencies along with this. Now, this is an inheritable disease. It is autosomal recessive. It can also, for other forms, it can be autosomal dominant. So sometimes you can predict this, but for many of these individuals, genetic counseling is an absolute must before they think about having a child of their own. Now, Duchenne muscular dystrophy, this is pseudohypertrophic disease. We see this onset a little bit later, not immediately at childbirth, but probably 2 to 6 years of development. Here we're going to see generalized weakness of the muscle, and we're also going to see the calves enlarge. This is mostly going to be from edema, not necessarily from hypertrophy of muscle. Now, whereas the Fukuyama was very aggressive, very rapidly growing, this disease is going to be much more slowly progressing, and it will affect the voluntary muscles. However, even though slowly growing, its survival rate is very rare into your late 20s. This also is an inheritable disease, but it is X-linked recessive. And so females tend to be carriers of this. When you think about it, if you're going to have a female that is going to express this phenotype, then the father is going to have to survive until reproductive age, to teens and 20s, which again makes this somewhat of a rare condition. Here we can see an older individual with a later onset, undergoing physical therapy, suffering from the muscle wasting in the legs, and this is an example of the Duchenne type of muscular dystrophy.